Hello students, today we are going to study the remaining part of the topic, that is modes of reproduction part 2. In this case we have studied many matter in this topic. Now let us go to remaining part. This is apomixis. What does it mean? Apomixis refers to the development of seed without sexual fusion. That is called apomixis. Now this apomixis, it is a fertilization. There is no sexual fusion even though development of seed takes place. For example, embryo develops without fertilization. So this is a sexual means of propagation. It is found in different crop species and it is very a common thing, and it is very useful in crop production or agricultural science and technology. There are 4 types of apomixis. First is parthenogenesis, second is apogamy, third is apospory, and last is adventative embryony. These are the 4 types of apomixis. Let us go one by one. Parthenogenesis, in this case, there is a development of embryo from egg cell without fertilization. Now this parthenogenesis is very important, where there are 2 types. One is haploid and another is diploid. This haploid parthenogenesis and diploid parthenogenesis. Now what happens in haploid? When the embryo develops from haploid egg cell, it is known as haploid parthenogenesis. Now in the plant where this kind of parthenogenesis takes place, embryos are haploid and sterile. For example, Solanum nigerum or nigerum, this is a very famous example. So this is found in some crops, but sometimes this embryo sac develops without reduction division, such that is embryo sac and cells are diploid. So here this is called diploid parthenogenesis, and for example it is given in the grasses like Tara sacum. In this grass it is found. It is also found in tobacco and rice and some other plants also. There are several causes of parthenogenesis, and in this case, why parthenogenesis takes place? For example, inability of pollen tube to discharge the contents inside embryo. Second, that insufficient attraction between male and female gametes. Third is that early degeneration of sperm. Fourth is very long style, short pollen tubes and slow rate of pollen tube growth, and stimulation of pollen in absence of pollen tubes, and incompatibility. Means because of various reasons, fusion between male and female gamete when does not take place, in that case parthenogenesis occurs. Second is apogamy. Now in this apogamy, what happens? The origin of embryo from either synergids or antipodal cells of embryo sac, that is called apogamy. Now here in this case there are again 2 kinds of apogamy, that is haploid apogamy and diploid apogamy. Now, if the embryo develops from haploid antipode cells, it is called haploid apogamy. But when the embryo develops from diploid antipodal cells, it is called diploid apogamy. Third is apospory. Apospory means what? In this apospory, first diploid cell of ovule lying outside the embryo sac develops into another embryo sac. That is called apospory. So here, without reduction, this thing takes place. So that's why it is called apospory. Now there are two types: generative apospory, somatic apospory. Now come to the fourth one, adventive embryony. Now this adventive embryony, what happens? Development of embryo directly from the diploid cells of ovule outside the embryo sac belonging to either nucleus or integuments is called as adventive embryony. So what happens here in this case? There is no production of another embryo sac as it happens in apospory. So this type of sporophytic budding, it is found in case of citrus and mango. Now this is the thing. Then come to another, that is a tissue culture technique. Now this tissue culture is a very important technique in agriculture because in less space and with less expenditure we can produce saplings on a large scale used for production of the plant or propagation of the plant. Now what happens? Let us see here in the diagram. So this start from here. Now from this plant, this is the plant. From this plant, a piece is taken. This is a small piece. This small piece is it can be taken from the leaf. Generally, it is called explant. Now this is used in nutrient media. So this is the media. For example, agar media is there. Different medias are there. Soil is a medium of plant growth. We have studied. So soil is also a media. So this media in this agar culture, media is a culture. This small piece of leaf is put. Then this leaf develops into a callus. Then embroid, it forms embroid. Embryo is formed and last product. So this is a plantlet. So this plantlet is formed. So this is the, in general, tissue culture technique. Now let us go in detail this tissue culture technology. This is based on a theory of totipotency. Totipotency is what? It is the ability of single cell to develop into whole plant. So when one cell gives rise to a complete plant, then that technology, that is called totipotency. So this is used in case of sugarcane also, particularly in banana. It is used in large scale. So here in this case, explant is used. Explant is excised part of plant, and this is grown in medium in a glass vessel, that is called in vitro. This is the medium is used in vitro, means in the glass vessel, and there is a prevention of all diseases. So prevention of these diseases takes place, and there it provides a particular cultural environment, and then after this formation of callus, it is transferred into this plantlet, transferred into the glass vessel. So small 1 or 2 centimeter long these plants are formed. Those are called saplings also. So then later on, they are kept in the vessels for sometimes for hardening, means for getting acquainted with the atmosphere. This is the protocol of tissue culture. Now this technique a little bit become different in different plants, and as per the kind of plant it takes place. So this tissue culture is also called micro-propagation. This is a micro-propagation technology, and it has become very invaluable contribution to agriculture for giving disease-free plants, quality plants, commercial plants, all these things. So this is very important. We have to remember the definition. Plant tissue culture is the technique of growing plant cells, tissues, and organs in an artificially prepared nutrient medium under aseptic and controlled condition. So this is a technique. What happens in this technique? Plant cell, tissues, and organs are grown artificially in nutrient medium. Now naturally plants can grow in the soil, in the nature, that is a different thing. But this is plant tissue culture is done in controlled condition, means in the laboratory. So very small plants parts are grown aseptically, means in a hygienic condition. It is done, nutrients are provided, and in a very controlled condition. It is done, controlled condition of light, controlled condition of humidity, then this temperature, all these conditions are controlled. So there are two types of plant tissue culture. One is in vitro culture or in vitro growth. Now what happens here in this case? Suspension of culture or explant is used for culture initiation, that is called embryo culture. So this is in vitro growth. Then here in this in vitro growth, whatever the plant part piece of a leaf is taken, that is called explant. Don't forget this word, explant. So totipotency technique is formed to grow, divide the cell, and the callus is formed and plant is formed. So this is a very important technique. It is called in vitro culture. Now this is essential. For this, what is essential? We require medium, vessels, glasswares, working tables, and hygienic condition. All these things are required. Now there are various techniques in tissue culture. Let us go to that technique. First, it is a micro-propagation. Actually, tissue culture is called micro-propagation. Now, what happens in micro-propagation? What is the purpose of this micro-propagation? Is to develop high-quality clonal plants. Clonal plant means a clone. What is clone? Clone is a group of identical cells. That is called clone. Now, because of this technique, we can produce the plant 100% as it was like the mother plant. So there will be no change in the quality of the plant. So then this is the micro propagation. What happens here in this case? Rapid and large scale propagation takes place. Somatic cell genetics. So here this is used for haploid production and somatic hybridization. Then another is anther culture. Now what is anther culture? What we are studying? Different techniques in tissue culture. Now in this anther culture, this is produced by haploids, means and doubling the chromosomes without going into series of back crossing. And it can produce the homozygous plants. homozygous plants are produced. So this is used by plant breeders. There another is embryo rescue. So when it is difficult to produce plant from seed, then this technique, embryo rescue technique, is used. And in this case, the seeds are surface sterilized and split open in aseptic condition, and the tiny embryo is excised and planted in a nutrient medium and grown. So in this embryo rescue, what is done, in the sterilized condition, they are split, embryo is removed and planted in nutrient and medium, and then plant is grown. Another is organelle transfer. Here, in some cases, this method is followed. It is a transfer of organelles or the cytoplasm into a new genetic background. This is another technique. Here, chloroplasts are transferred, including nucleus, that is also transferred. Another is transgenic plants. This is used for expression of mammalian genes or plant genes for various species. It is very useful in plant engineering, means we can produce the disease resistant, virus resistant, insect resistant plants. So this is a transgenic, means we change the genetic pattern of the plant. Then methodology involved in plant tissue culture. What is the methodology? This process, in this process, use of small piece of given plant tissue. This is taken. This tissue is used, it is in an appropriate medium under sterilized condition. So here plant tissue culture, let us see using a diagram. So this diagram, have a look at diagram, steps in tissue culture method. From top to bottom, let us go from top to bottom. Glassware, media, explant. So in this glassware, media is used, and in this media, explant is a micro piece, very small piece of leaf or another part of the plant is used. This glassware is already sterilized in using autoclave or dry heat. The explant is also surface sterilization is done. Then this media, what is this media? Media is also, in this media, gibberellic acid is allowed, component is used, and pH is adjusted. It should not be high acidic or high alkaline. Then in this media, there is a agar, that is called agar-agar solution. So this is a solid or semi-solid form. It is poured in the vessel. Then this glassware is wrapped in brown paper, and in this glassware this media is poured, and then laminar air flow is there. Then this explant, a piece of leaf, is added at this position. Then this growth culture is added, and then this explant differentiation takes place, means growth takes place, callus formation takes place, and then it can be transplanted into another test tube if necessary. The subculture can be used. So this is the this is the plant tissue culture. Now this is general procedure it is to be followed. Now medium, how to prepare this medium? For example, agar-agar medium. What is this medium? Medium is nothing but the nutrients. We provide nutrients in an artificial way for growth of the cell into the callus. So it is a mixture of, it is a distilled water. In this case, some sugar or sugar mixture is taken, sodium hydroxide is added, or hydrochloric acid is used to adjust pH. Now this depends upon the tissue culture we are using for each plant. Then agar is added in this mixture. A little heat is given and stirred to dissolve that agar. Then it is cooled. The warm medium is poured into polycarbonate tubes up to 4 cm depth. Then lid is put on the tubes, and using a pressure cooker, or it is sterilized for 20 minutes. So this becomes a medium. Then plant preparation. So this, just we have studied this media.